Hello, my name is Rma Polce, and today we're presenting Lipocalin 2, the new possible target for therapeutic intervention in cancer cachexia. The motivation for this project first stems from the cancer cachexia cycle, which is characterized by multiple metabolic process changes, including increased systemic inflammation and decreased protein synthesis and quality of life. The next part of the motivation for this project is the multifaceted effect of Lipocalin 2 in the body. Lipocalin 2 affects things like the kidney, the liver, and the brain. Cancer cachexia, CACs, is characterized by loss of skeletal muscle mass and adipose tissue, as well as anorexia and systemic low-grade inflammation. Around half of all cancer patients will develop a form of CACs, with CACs being extremely prevalent in lung cancer patients, about 60% development, and 20 to 30% of all lung cancer patient deaths being related to CACs. CACs patients also become more prone to experiencing comorbidities such as chemotherapy-related toxicity. Lipocalin 2, LCN2 expression increases highly in response to cellular stress. Expression is also correlated with inducing the browning of white adipose tissue, and expression in cancer patients has been linked to worsen prognosis and cachexia, as well as increased tumor metastasis and growth. From here, I derive my problem statement. Around half of all cancer patients will develop a form of cachexia, with development being extremely prevalent in lung cancer patients. Despite this, there's no FDA-approved treatment at the moment. This allowed me to propose the question: what is a possible new target for therapeutic intervention in cancer cachexia? This allowed me to create my hypothesis statement. Lipocalin 2 is a promising new target in therapeutic intervention in cancer cachexia due to its multifaceted roles in tumor microenvironment, appetite control, and energy expenditure. First, mice were genetically engineered using the LISC system to create LSL, KRAS, G12D+, LKB1 flux mice. These mice had KRAS, a tumorigenesis gene, activated, and LKB1, a tumor suppressant gene, removed. Next, data was collected from the liver, the gastrocnemius, the tumor, and the white adipose tissue, and weighed to generate bulk RNA sequencing data. From here, the RNA sequencing counts were normalized to FPKM values, analyzed using online softwares. Here's the model for genetically engineering the mice. So first, the mice were bred with a genetic change. The LISC system was injected, which then activated these changes in the lungs, causing some mice to develop cachexia and some not, making them easily comparable. So first is the expression of LCN2 in the liver of the cachectic mice. The red color indicates extremely highly upregulated genes in the perturbation or cachectic mice. So here we see LCN2 as part of this category with a log fold change of 2.9 and a p-value of 0.0176. Here we have the expression of LCN2 in the gastrocnemius or muscle, and we see it follows the same trend as the liver, being extremely highly upregulated with a log fold change of 1.8 and a p-value of 0.0096. Here we have the expression of LCN2 in the serum of the cachectic and non-cachectic mice. So first we see in the non-cachectic mice that there's very little blotting of LCN2, indicating that it is not highly present. However, in the cachectic mice, we see much darker blotting, indicating that it is upregulated. Here we have the expression of LCN2 in the white adipose tissue of the cachectic and non-cachectic mice. So we see here in the cachectic mice, when compared to non-cachectic mice, that there is not extremely highly upregulated levels of LCN2. However, it is right on that barrier with a log fold change of 3.12 and a p-value of 0.000062. Here we have immunohistochemistry images of the white adipose tissue of cachectic and non-cachectic mice. So first we see in the non-cachectic mice that there's little evidence of browning and only few deposits of beige adipocytes. However, in the cachectic mice, we see widespread browning and large deposits of beige adipocytes. Here we have the expression of LCN2 in the tumors of cachectic and non-cachectic mice. So first, in non-cachectic and cachectic mice, we don't see much variance in levels of LCN2 in the tumors. However, it is important to know that it is highly upregulated in both. So overall, LCN2 is upregulated in the serum, the liver, the gastrocnemius, and the white adipose tissue of cachectic mice. Cachectic mice experience more browning of their white adipose tissue than non-cachectic mice, and that within the tumors, LCN2 is robustly expressed. Some of the limitations of the study first include we do not have data from every tissue. Since LCN2 affects so many tissues in the body, it could be important to see what is going on in other tissues and how it is playing a role there. Next, this is only tested on mice with lung carcinomas. Since each cancer varies, it could be important to know this might not apply to all cancers. Even further, mice are not a perfect model for human carcinomas and conditions, so there could be some variance there. But overall, the hypothesis that LCN2 is a new promising target for therapeutic intervention in cancer cachexia is supported. Some future research could first include using the LISC system to eliminate LCN2 from the white adipose tissue as well as the whole body. Next, investigate MC4R dependent suppression of appetite by bone-derived LCN2 and serum-derived. So this was seen in a pancreatic model, so it could be important to see what's going on in the NSCLC model or this model. And next, we can also introduce an antibody that will neutralize the effects of LCN2 on the tumor and the tumor microenvironment. From here, some future expected outcomes first include reduced energy expenditure due to impaired browning of adipose tissue, increased appetite due to lower levels of circulating LCN2, and a reduction in tumor growth due to the lack of iron needed for the epithelial mesenchymal transition. Thank you.